Good morning. I am in Alcântara. I'm from Federal University of Maranhão, Brazil. I will talk about the all recent work accepted to publication in a special issue about clay minerals and the health applications in clays and the clay minerals journal. I would like to apologize for my webcam. In the last few days, it shows some problems. First of all, I would like to thank this invitation, especially to Professor Eduardo Rizhitsky, to whom I have a great respect and admiration. It's a pleasure to be here today to presenting this work entitled Layered Clays as Versatile Nanodevice to Drug Delivery Systems. This work involves a student of the master degree and is a resulting of the partnership with Vera Constantino from the University of São Paulo. The outline of this talk consists in a brief introduction where we talk about the clays and the pharmaceuticals, with focus on clays, we focus on montmorillonite, and the drug delivery, the bionanocomposite based on clays for pharmaceutical purposes. Following the objectives, the methodology used, the results, and the conclusions. Among the most diverse of thousands of clays minerals now, in the pharmaceutical industry, only about 30 minerals are used, and the most of them are synthetic minerals. Clay minerals are notable exception because their synthesis is difficult and expensive, and therefore they are used as mineral. In this case, the clay minerals in the pharmaceutical purpose can act as active ingredients as anti-acid, antibacterial, or excipients for different pharmaceutical forms, such as biphasic dosage, semi-solid or solid forms, where the clay can act as a stabilizing agent, binder, diluent, release retardant of drugs. Among of the clay minerals that can be used for the pharmaceutical sector, the montmorillonite is the one of the most used clay minerals in the preparation of the large variety of the functional and the structural hybrid materials. These clay minerals is belonging to the phyllosilicate family, the smectite, and shows cations in the interlayer region in order to compensate a negative charge of the layers due to isomorphic substitution and imperfections of the crystal lattice. These cations can suffer ion exchange reactions, allowing the accommodation of ions or molecular and the macromolecular species in the interlayer spaces. So these cations can be replaced by alkyl ammonium ions, for instance, in order to obtain organoclays, or dyes, or polymers, or drugs. In the last case, the case of the drugs, the montmorillonite is very interesting vehicle because shows good absorption ability, low toxicity, abundance, and low cost. In this sense, the montmorillonite is used as a carrier for a lot of drugs such as tetracycline, amoxicillin, metformin, metronidazole, neomycin, isoniazid, olanzapine, for instance, where the intercalation of the drug in the interlayer space enhancement the chemical stability and the dissolution properties of the drug, and consequently decrease the undesirable side effects due to the modified release to reach optimized concentrations in the organism. However, the advantages of the drug intercalation can be enhanced due to association of these intercalations compounds with biopolymers, such as polysaccharides or proteins, to form bionanocomposite materials that act as hybrid devices for drug administration. Besides acting like a chemical and mechanical barrier to protect the intercalated bioactive species. Biopolymers are naturally degradable macromolecules and the biocompatible macromolecules. So it's very useful for drug delivery applications. Um, Among these biopolymers, from the point of view of biopharmaceuticals, the gelatin protein and iota carrageenan polysaccharide show interesting and important features such as no toxicity, high biocompatibility, and the hydrophilicity, and the possibility of to be processed as film, beads, or gels in order to attend the different vias of the drug administration. With the basis of this premise, the main objective of this work is the development of the porous bionanocomposites based on montmorillonite layered clay and iota carrageenan gelatin biopolymer blend as drug release system. For that, we have the preparation, the pro-montmorillonite drug intercalation compound, and their respective bionanocomposites processed as porous beads through a freeze drying process. We also evaluate the influence of the montmorillonite clay in the material under the freeze step in vitro tests of the drug delivery in conditions that simulate the gastrointestinal tract. And with that, we expect to contribute to the knowledge of clays-based bionanocomposite materials for pharmaceutical sector. The ciprofloxacin antibiotic drug was the drug model chosen for this work. This molecule is widely used for human and veterinary infections, but show also diverse adverse effects for for the body. The methodology used in order to obtain the montmorillonite ciprofloxacin hybrids, the method used was ion exchange reaction, where the suspension of the sodium montmorillonite it's added the solution of cationic ciprofloxacin prepared in pure water, at a pH of 3.5 during 1 day. Okay. After that, the suspension is centrifuged and the solid is isolated and obtaining the montmorillonite ciprofloxacin intercalation compound. For the bionanocomposites preparations, a mixture of solution based on gelatin and iota carrageenan dissolved in hot water is prepared, obtained in this case a blend solution. Okay, this blend solution it's a that ciprofloxacin drug in the free form or intercalated or previously intercalated on montmorillonite in the same concentration, in the same amount of ciprofloxacin, 0.1 gram approximately. Obtained in this case the bionanocomposite solution. In order to favor to cross-link process, the system is a dead drop-wise and 50% of the calcium chloride solution, and obtained in this case the bionanocomposite beads. These fresh bionanocomposite beads were separated in two parts. The first one, the first fraction, was frozen by conventional freezing for 1 day, while the second one was frozen by immersing in liquid nitrogen, and both fractions were subsequently freeze dry, obtaining in this case the porous bionanocomposite beads. Uh, uh, the results. In order to investigate the intercalation, the formation of the intercalation compound, these materials was evaluated by XID measurements. In this case, it's observed the 001 reflection from pristine montmorillonite is shifted towards lower degree to theta, displaying in this case a new basal distance of 1.98 nanometers, which indicates the intercalation of ciprofloxacin molecules between the layers of the montmorillonite. Considering the molecular dimensions of the ciprofloxacin, it's expected a demolecular ciprofloxacin is slightly tilted arrangement with respect to aluminum silicate layers, with opposite directions of the piperazine groups due to the cationic ciprofloxacin electrostatic attraction with the clay surface. The amount of the ciprofloxacin incorporated the hybrid material was calculated by elemental chemical analysis, showing 76 megaequivalent per 100 of clay. Considering that the cation exchange capacity of the sodium montmorillonite is already 93 milliequivalent per 100 of clay, this value indicates that the exchange reaction was not complete, and the presence of a small amount of sodium ions is expected in the interlayer space. The intercalation process was also investigated by infrared spectroscopy, where it is observed that besides the bands from the clay mineral in the intercalation compound, the montmorillonite ciprofloxacin spectrum, is observed also a new band at 1709 centimeters minus 1, assigned to the carboxylic group, which indicates that this drug is protonated and corroborates the presence of the positively charged ciprofloxacin intercalate into montmorillonite. In addition, it's observed also a displacement of 10 centimeters minus 1 in the band uh, in the band at 1373 centimeters minus 1 in freeze ciprofloxacin towards higher wavelength values in the hybrid materials, which suggests the protonation of the amino groups in the piperazine moiety. These results indicates the establishment of the electrostatic interactions between negatively charged surface of the silicate and the drug in its protonated form. The results of obtained by infrared spectra can be also confirmed by solid state NMR, 30 carbon NMR spectra of the hybrid material in comparison to that free ciprofloxacin in red color here. In this case, the resonance of the ciprofloxacin, the pure ciprofloxacin at 51 and the 42.3 ppm, which correspond to the interactions of the carbon atoms bonded to nitrogen, are selected shifted to 51.7 and 41.8 ppm in the hybrid material, which indicates the possible change in carbon nitrogen binding energies. Once the ciprofloxacin, the montmorillonite ciprofloxacin hybrid material was characterized, this intercalation compound is added at the biopolymer matrix, and the beads, the poro beads, is prepared by two different methods, by liquid nitrogen or conventional freezing. The encapsulation efficiency and the loading of the drug in these systems were also evaluated. Were observed that approximately this observed three three times more increase of the drug loading and encapsulation efficiency of these materials based on intercalation compound compared to the analogous materials um, prepared with free ciprofloxacin in both cases. This fact could be associated with the possibility of the slower diffusion of the some drug molecules from the interlayer region of the silicate, contributing to the good affinity between the montmorillonite and the biopolymers used in the present study. Um, However, it is important to highlight that there is not significant differences in the drug loading and the decapsulation efficiency values between the bionanocomposite samples frozen through conventional or liquid nitrogen freezing methods. But this behavior is expected since this drug incorporation process is performed prior to freezing step. Then, this process should not have any influence in the amount of the encapsulated drug. Considering that the apparent density can be influenced by the freezing process and consequently the release of the ciprofloxacin from the beads, the property was also studied. All the bionanocomposite beds frozen by liquid nitrogen show higher apparent densities compared to the beads prepared to conventional freezing, probably due to the cellular structure composed mainly of small and few pores. It observed also that materials based on montmorillonite ciprofloxacin intercalation compound showed more accentuated differences in the values of the apparent density, which indicates that the incorporation of this hybrid material in the biopolymer matrix can directly influence the process of the cellular structure formation of the final material. Also, should be considered that the presence of the hybrid in the biopolymer blend contributes to increase of the specific mass of the system and consequently increase the apparent density of the bionanocomposite materials. um the bionanocomposite materials, sorry. The surface of these bionanocomposite materials was also observed by a scanning electron microscope. In the in the case of the beads prepared by liquid nitrogen, it's observed a more compact morphology with domains of isolated small pores is observed. In contrast, conventional freezing is observed a porous surface and slightly larger pores throughout the material. This differences in the porosity of the beads can be attributed to growth rate of the ice crystals. In the case of the liquid nitrogen, it's enough. At this temperature, it's enough. It's low enough to allow the rapid freezing of the water molecules, not allowing the indiscriminate acid growth. Conversely, in the conventional freezing, the ice grows slowly, produces larger crystals, and consequently a large porous structure after removal of the solid phase by freezing dry. This effect is usually observed in the nanocomposite materials, where the porous structure resulting for the elimination of the ice crystals it's more evident in materials prepared for conventional freezing than in those prepared with liquid nitrogen. In addition, in the case of the bionanocomposite materials based on montmorillonite ciprofloxacin, the incorporation of the intercalation compound in the biopolymer matrix may help the forming process, acting as nucleate acids at the same time that can modify the mechanical and the physical properties of the final material. Another another property um, evaluating this study was the water uptake using the ionized water. In this case, the water uptake showed different profiles um, depending the freezing method employed in the preparation of the bionanocomposite bits. In general, the presence of gelatin seems to contribute to enhancement of the water absorption, independent of the method of the freezing used. This enhancement of the water absorption is probably due to the highly hydrophilic character of this protein. Uh, Also is observed the presence of the montmorillonite ciprofloxacin in the beads show lower values compared to analogous biopolymer beads, which indicates that these systems based on intercalation compound offer additional stability in aqueous media. Um, when compared these semi bionanocomposite systems in the compared systems in their different freezing methods, it's observed that those submitted to conventional freezing showed higher values in the water uptake in pure water, which can be attributed to the porous surface formed by the freeze method. Um, the freeze method, the conventional freeze. And finally, the in vitro ciprofloxacin release profile was investigated. Also, the cumulative percentage, in this case, the ciprofloxacin released as a function of the time was determined by in vitro drug release assays, where the bionanocomposite bits frozen in conventional freezing or liquid nitrogen were kept in contact with simulated stomach and intestinal fluids at different pH values for around 8 hours in order to simulate the different steps of the gastrointestinal tract as in vivo process. For comparison purpose, the ciprofloxacin pressed as a tablet was also investigated. Mm, in both cases, it's observed the free ciprofloxacin tablets is completely dissolved at pH 1.2 in the acidic pH after 30 minutes of assay. Um, In contrast, when this ciprofloxacin is intercalated, in the montmorillonite, in both freezing methods, the presence of this um, montmorillonite ciprofloxacin delay the release about uh, um, 10% in the release of the ciprofloxacin when compared to montmorillonite ciprofloxacin hybrid on uh, only the montmorillonite ciprofloxacin only, and 40% in relation to the material without the intercalation compound. But interesting observed also that the intercalation compound by itself showed also a modified release profile of ciprofloxacin. It's very, very interesting. But when this intercalation compound is incorporated to the biopolymer, this release it's more slowly. It's more slowly. The conventional freeze, comparing the freezing methods, the conventional freeze uh, seems induced cell structure. The conventional freeze induced cell structure seems to promote the release of the encapsulated drug compared to the liquid nitrogen due to the three-dimensional arrangement format and the higher water absorption as demonstrated in the water uptake studies. The conclusions of this work, the intercalation of the ciprofloxacin antibiotic drug into montmorillonite was carried out and by ion exchange mechanism, which was confirmed by XRD measurements. Spectroscopic analysis indicate the stabilization of the ciprofloxacin into intercrystalline space of the clay, where the electrostatic interactions between the drug and the decationic form and the negatively charged of layers of clay take place. Bionanocomposite bits were obtained by incorporation of montmorillonite ciprofloxacin hybrid into a biopolymer matrix composed by iota carrageenan and gelatin, using liquid nitrogen or conventional freeze to assist the drying process. The bionanocomposite beads prepared by freezing in liquid nitrogen show more gradual drug release compared to the beads prepared by conventional freezing due to different porosity in these structures, as indicated by same images and the uptake studies. The presence of the montmorillonite ciprofloxacin in the material seems to favor the formation of the larger pores, where the hybrid material acts as nucleation seats, as observed by same images. The results reported here indicate that the association of ciprofloxacin intercalation compounds with a iota carrageenan and gelatin can act as hybrid porous drug release device, where the release of the active principle can be modulated depending of the freezing method employed in the preparation of the material. I would like to thanks to the professional support provided by CNPq and the FAPEMA and the institutions Federal University of Maranhão and University of São Paulo, and special thanks also to my students. And I'm available for any question. It's a pleasure to answer any question by me. Thank you so much.